A couple of years ago, I went to a totally normal party. It was put on by the well-muscled brain explorer, Brian Johnson. He held the event at this house in Venice, California. Billionaire Patrick Collison was there, and so was his smile. The guests all got together, donned Bluetooth headphones, and listened to music written by an AI and performed by American Idol contestant Taryn Southern. Oh, and you could also take a whiff of an extinct Hawaiian plant, because some people who play with DNA had found a way to bring its smell back to life. The folks who did the DNA manipulation work at Ginkgo Bioworks. The company is perhaps the world leader in the field of synthetic biology. Earlier this year, Ginkgo raised a staggering $2.5 billion on its way to being valued at $15 billion. To find out what $2.5 billion buys you these days, I went to Ginkgo's headquarters in Boston, Massachusetts. For it is here. Or make that here, that the company is building out a DNA superhighway. Ginkgo was founded back in 2009 by several big brains from MIT, including its CEO, Jason Kelly. The founders wanted to develop ways to treat DNA more or less like software code. You find an organism with an interesting property, snip out the DNA behind that property, and then shove the DNA fragment into something else. The big promise behind synthetic biology is that we'll find a new means to harness nature and bend it to our will. We'll be able to manufacture things like chemicals and fuels and even physical objects in ultra-efficient ways. To be clear, we're also talking about eventually getting into Jurassic Park mad scientist-style stuff. And this grand experiment could end up with you being a reptile's breakfast.
The core idea behind the company was inside of every cell, every bacteria, plant, animal on planet Earth, is digital code in the form of DNA. Right, so it's A, T, C's, and G's, not zeros and ones like you'd see in a computer, but you can read that code with DNA sequencing, and importantly, you can write that code with DNA printing. And this is, like, for a bio nerd like me, this is magic. You literally go on a computer, you type A, T, C, G, 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 you hit print, and out of machines like in this facility or the ones next door, comes a piece of DNA that's exactly what you ordered. And then you can put that into a cell. And our idea was, if you can read and write code, and you have a machine that'll run it, which is sort of how we thought of a cell, you're programming, just like you'd program a computer. And so Ginkgo's business is basically programmer tools so that other folks can develop new programs for cells.
This core synthetic biology principle of treating DNA like code is simple in theory. In most cases, scientists will grab the DNA they want and insert it into something like a bacteria or yeast. Then they create lots of copies of the organisms in much the same way as we make beer. They feed the organisms sugar and nutrients and grow them in a vat.
So I'll give you an example. There was a flower that went extinct in I think it was like the early 1900s in Hawaii. But before it went extinct, it had gotten collected by scientists and then stored, and it was at the Harvard Botanical Museum. And so we collected some of that sample, brought it back here to Ginkgo, put it through our DNA sequencer, so we read the DNA code of that extinct flower. We found the part of the DNA that encoded for the scent. So we would take those 6,000 letters of DNA and move them into a bacterial genome like E. coli. So how do you do that? Well, you have a special enzyme that basically can find a location on the genome. It looks for A, T, G, Gs, whatever one is on the specific code it has, and it cuts. And then it opens, and then you add in those 6,000 letters from the extinct flower genome. How did you get them? Well, you read them, you had them on the computer, you put them in ATCG, hit print, and you printed a piece of synthetic DNA. So now you add that in to where you cut, and then you patch it up a little bit, grow the cell. And instead of just doing the normal swim around bacteria thing, it also now makes this fragrance.
Yeah.
And so we actually resurrected the scent of this extinct flower as a project that is actually showing at a few art museums to kind of bring it back.
To see some of this process in action, I went to visit Sarah in one of Ginkgo's labs. Okay, so you've made a microbe, you've engineered it with a specific bit of DNA to give it a quality that you're looking for,
Yep.
and then you guys are both making those and checking that they're coming out right.
We're testing a lot of different engineered strains to help us build our code base to understand which of those pieces of DNA are able to achieve the desired outcome.
Okay, and so, you've got these cells over here. Some of them have stuff on them, the white, and some of them don't. So I assume the ones with the white is kind of ones that have worked, and that's what you're looking for.
Yeah, so each of those individual segments is a particular engineered strain that we are trying to isolate in order to then test downstream. The segments that have the individual white circles, which we call colonies, have successfully taken up that DNA.
So this would be like the beginning stage of you get what you want, and then you can grow it in bigger quantities.
You can study
after.
it.
Yeah, okay.
So this is the very start of that kind of pipeline.
What Ginkgo really wants to do is automate all of this and make meddling with DNA fast and cheap. To do that, you need a factory. This is perhaps the most modern DNA assembly line on the planet. Molecules go in at one end of the conveyor belt, and robots take care of all the processes needed to refine them into something useful.
So this is a magnetic propulsion system that has individual carts, and the robots or the humans will load samples onto these carts, and then the system will know which destination they need to take them.
So a human comes here and puts a sample, feeds it into the
Exactly.
system.
There you go. And now you can press the button over here.
Okay. And then off it goes. And the green robot is
Now
going
you
to
can
grab
see
it.
the green robot pick it up, and it will place it on this second station there, and this is when the yellow robot will come into play. The little yellow robot will remove some of the samples, add controls in their place. The tube rack then goes to the next processing station, which is a centrifuge work cell here.
Okay.
Spin it around, essentially to try to remove bubbles or anything that might have formed during shipping of the samples.
Okay. I think our sample's off again.
So now we're sealing the plate, and it can go over the rest of the process.
It all ends with an incredibly sophisticated trash can.
These specialized trash work cells have these specialized carts that can weigh the amount of trash that is in each of these bags to know when they are full. And finally, the robot can actually place the trash at random positions around the bag to make sure that they're not always stacking up in the
Like a
center,
tower.
making very large towers that can eventually break the system.
And does a human have to take this out at the end?
This is the last step where the human is still in the loop.
Step forward, one helpful TV host. I remember around 2008, it was getting easier a bit to manipulate DNA, and we could see where things were going, but people were already saying, in a few years, you will program a cell to make a chair, and we won't have chair factories anymore. We'll just grow them. And so how much of that vision do you still believe in, and how close are we to this idea that we're gonna just be making kind of like fantastic new things out of biology?
I think it's all coming, yeah.
You do?
Yeah. The reason is, I cannot emphasize this enough, people take biology massively for granted.
Yeah.
Every other engineering field was designed by humans from the bottom up. Like, we know every little bolt in that Ford car, every little like gate in that semiconductor chip from Intel. Well, biology is ridiculously advanced And we're just entering the era of being able to program it to do new things. We know it's capable of big things.
Yeah.
The only limit is human understanding to be able to program it. But we know if we learn, it can do this stuff because it's already doing it. It's making our food, it's cleaning our water, it's making the air, you know, we're made out of it. And the one magical feature of it, and it is a miracle, is it's programmable because inside of it is digital code that we can, as humans, read and write.
Ginkgo does go after some weird stuff. This dog, for example, used to be a teacup poodle just last week. And clearly people here really do dream of doing things like resurrecting the dinosaurs. But as with any new technology, I wanted to know how synthetic biology would benefit me, like right now. And so I took what we'll call the Blood Elevator Express, a couple floors down to another startup, Motif Foodworks. Motif is sort of a research and development company for food. It uses synthetic biology to make food taste better. Today, I'm here to eat a burger, only no cow was involved. Motif teamed with Ginkgo to take the DNA code for hemoglobin, plop it into a yeast, and then grow what amounts to a vat of faux blood. This fella here is adding the blood to some soy, throwing in some seasonings, and then, voila.
Thank you so much. Well, should we try it while it's still hot?
Absolutely, that's the best way.
Okay.
Good.
It's burgery, for sure. And it has that, like, on the char and all that. You
The
get
char.
that- Yeah.
The bite quality is much better. I would say that old generations of plant-based meat, the first bite you might get this crust on the outside, like you see here.
Yeah.
But the second bite would be more mushy. The inside would not have texture. So look at all the effort. I mean, that pretty much looks like what you might see in a burger.
Yeah.
You notice all the pieces, the way it crumbles.
The other thing I noticed is there's definitely that umami kind of note on
Our
there.
ingredient we call hemami, and it's essentially the molecule in the muscle of a cow that makes beef red. It makes it give you that beautiful serumy, bloody flavor. It catalyzes a whole bunch of the chemistry in there. And that is an amazing piece of technology that Ginkgo Bioworks created for us. Ginkgo was able to look at 300 different muscle proteins from different species. Mini sperm whales, one that comes to mind,
Okay.
tuna, pork, chicken, a whole series of different birds. And they were already sequenced, so it wasn't like we took any animal material.
Yeah. When they did the sequences, it was for...
Specific muscle protein, a class called myoglobins.
Okay.
Ginkgo got us the ability to look at all the different properties, stability, taste, flavor, color. And then once we honed in on the ones we wanted, we asked them to optimize the yeast to kind of turn that into a mini factory so that we can make it and so we can have the great taste of beef without using any animals.
To put a fine symbolic point on how all of this stuff gets made, I felt it essential to go to a nearby brewery with Jason.
I thought being in a brewery was...
Yeah, ideal, right? They use yeast, but like no commercial beer today is made with like genetic engineered cells. Have you ever had a non-alcoholic beer?
Yeah.
It tastes like shit?
Yeah.
Well, that's because it's getting rid of the alcohol, you're like pulling off all the flavor components. And so, could you make a yeast that doesn't produce much alcohol, that gives you all the taste, so you end up with like a alcohol-free beer that actually tastes good? Brewing is an industry that has like biology so intrinsic to it that it's actually an opportunity to like figure that out and do it correctly. We've worked on the brewing industry. I think it's going to come around pretty soon, actually. Yeah. Oh, thank you. Oh, look at that.
Thanks. Cheers, man. To yeast.
Yeah, to yeast. Oh, that's good.
I wanted to go back just like a little bit. Where'd you grow up?
I grew up in Florida.
In Florida.
Yeah, South Florida.
And what was
Yeah.
your family as like kind of size family or?
We weren't like a family of PhD. I was the first person to get a PhD.
Yeah. But were you even like in high school and everything? Was
High school, I
science your
got
thing?
in. Yeah.
Yeah.
I honestly, I think what it was I was, 12, 13 years old when Jurassic Park came out.
Yeah.
And I was a big sci-fi nerd. I was reading books. Like I read all those Crichton books. I loved that stuff. And I think I fell in love with it then.
What kid wouldn't dream of playing God? To that end, I wanted to know when we'd get past perfumes and fast food blood and get to the good stuff, the freaky stuff.
When I was talking with the guys that helped make the show, this was one of the lingering questions that we still weren't sure about. I can get the enzyme to cut and paste DNA. I can get it to make the myoglobin, right?
Exactly,
but then, why can't you grow an arm? This is what we kept talking about in the car. It's like, why can't you put in the code for the arm then? If you can put in the code for the protein, why can't you do the whole thing?
So it's a great question. And this is like me when I got to MIT.
Yeah.
I'm like, I thought I would be making arms or something, right? And the short answer is we don't understand the specific sequence of DNA, how it affects the cell. It's just because we didn't design cells and they're complicated. And so if you put in some new DNA that was like, I think this DNA will make an arm, it just wouldn't, right? But it might make some other deranged thing. But it's like salamanders, the freaking salamander grows arms, but humans don't. And so it's just a programming problem between here and there, but we know the substrate is capable of it.
Obviously, everyone thinks about climate change all the time. As far as, and I don't want to give people false hope on any of this stuff, but something that's just gobbling. Why can't we just gobble carbon and shit out happiness on the other side? Where is that?
I'll give you a real one. I'll give you a real project. Okay, so the project we're doing, we have a $100 million joint venture with Bayer Crop Science.
Yeah.
Okay. Synthetic nitrogen fertilizer.
Yeah.
Okay. The way you make it, you basically pull nitrogen out of the air. You're basically going to take that nitrogen, you're going to combine it with hydrogen to make ammonia, which is fertilizer.
Yeah.
Where do you get the hydrogen? Oh, burning natural gas.
Right.
Okay, so you burn natural gas. About I think it's 4% global greenhouse gas is going to freaking fertilizer. But hey, we get to eat, you know, right? We need fertilizer, right? Okay. Well, there are certain crops like soybeans that don't need much fertilizer. Why? They have microbes in their roots. They're literally pulling nitrogen out of the air.
Nature doing all the work.
It's like you plant a legume and it re-fertilizes the soil. So we can go find the code that produces fertilizer for crops in soybeans.
Yep.
We can take microbes that are friendly with corn, install that code, and then a business that Bayer already has, you do a seed treatment. You basically coat the seed in those bacteria, and when you plant it, they grow along with it, and they produce fertilizer for the crop.
Yeah.
That's the project. So that's one where if you actually succeeded, not only is it a $70 billion market, it's a 4% global greenhouse gas reduction. Yeah.
No, that's cool. But but nothing's just out there gobbling CO2 out of the air yet.
Well, trees.
Yeah.
Yeah. That's an amazing machine. And it is when you fly on an airplane and you look out the window, what do you see? Trees. Right? That is real scale. Okay, none of this crap like our auto plants and chip fabs. It's a joke. Right? The only real scale on the planet that's actually making anything is biology. The only thing we're good at making is CO2 and fucking things up. Right?
To Jason's point, humans do have a tendency to run amok when given new toys. And this could all go bad. Jurassic Park bad. That said, synthetic biology is the area of technology that excites me the most. The stuff bubbling away in these jars and flowing through these tubes represents our best shot at fixing many of the messes we've made, and at doing so many other things better in the future. It's certainly not communing with nature in any traditional sense, but it's an area where humans can meld science and creativity in previously unimaginable ways. In DNA, there is hope.